Today we pick up in chapter 4, and we're actually going to start looking at different parts of the cell, specifically different organelles of the cell. Now, the first grouping of organelles that we're going to look at are going to be membrane-bound organelles. And so membrane-bound organelles are going to be surrounded by some sort of membrane, and it allows them to perform their activities essentially in an isolated environment since they are surrounded by a membrane. The first membrane-bound organelle that we're going to talk about is the endoplasmic reticulum. And when we look at the ER, which is what it's short for, endoplasmic reticulum, we're looking at an extensive interconnected membrane network. And so the ER is really going to vary in shape, but it has one continuous lumen. And when we look at the term lumen, you really should think of a kind of a hole or a passageway. So if we think about the small intestine, how it's kind of this long extended tube, the lumen is basically the hole that food is able to pass through. And so again, with the ER, we have this continuous hole that's passing through this kind of extensive network. It's going to extend from the nuclear envelope all the way to the plasma membrane. And so we're going to find that it is actually continuous or extensive of that nuclear envelope. It composes about half of the membrane within the cell, and it's the point of attachment for our ribosomes. And when we have ribosomes that are attached to the ER, we call it rough ER. The area of the ER that does not have ribosomes is called smooth ER. And we'll talk about different parts of those or different types of those as we actually get into specific examples. So if we look at the ER then, you'll notice that here is our nucleus, okay? Around the nucleus, we have our nuclear envelope, okay? And you'll notice that we have this ER, and you can see like essentially right through here, we have this ER that's basically an extension of what we would call a nuclear pore. And so that nuclear pore opens up into the ER, and then we have this lumen or this opening within the endoplasmic reticulum. And so we have this kind of extensive network of tubes and areas. Again, two different types. We have a rough ER and we have a smooth ER. The rough ER you can see here are studded with ribosomes. That's kind of what's indicated by those little yellow dots there. And then we have the smooth ER. And the smooth ER, you'll notice, still has these kind of holes or these lumen, this network area. But you will notice that it does not contain any ribosomes. One of the major functions that we're going to see with the endoplasmic reticulum is it is going to be involved with protein synthesis. And we'll see that a little bit more in detail when we get to that point of this chapter. So our rough ER is involved in the protein production by the ribosomes. And so the proteins that are created by ribosomes are actually inserted into the ER. At this point, the original structure of the protein is going to be changed, and it's going to be transported out in enclosed membranous sacs. These membranous sacs typically are what we call transport vesicles, and they're going to shuttle proteins from the rough ER lumen to our Golgi apparatus, which is going to be one of our next organelles that we discuss. Now, the smooth ER typically is involved in a little bit more metabolic processes. And they're going to vary by cell. A few of the functions of smooth ER are going to be involved in synthesis, the transport, and the storage of lipids and fats. It's going to be involved in carbohydrate metabolism. And it's also going to be involved in the detoxification of drugs and poisons, okay? One of the other functions that we'll see is we'll actually see this when we get into muscle contraction, is it's essentially going to be a storage for calcium. So our next one is the Golgi apparatus. The Golgi apparatus is composed of cisternae, which essentially are elongated sac-like membranous structures. They are going to exhibit polarity. And so when we look at the polarity of the Golgi, we have a cis face and a trans face. Now the cis face is going to be proximal to the ER, so it's going to be closer to the ER than what the trans face is. The trans face is going to be distal from the ER, so it's going to be a little bit farther away. It's actually going to be closer to the plasma membrane. And the major functions of Golgi apparatus are going to be modification, packaging, and sorting of different proteins that are created and transported from the ER to the Golgi apparatus. Okay, so modification, packaging, and sorting of proteins. It does form secretory vesicles. These secretory vesicles are going to be a part or oftentimes become a part of the plasma membrane, while others simply release the contents outside the cell. Okay, so as these vesicles move towards the plasma membrane, they can actually join, be a part of the plasma membrane. And then again, some of them are going to release the contents outside the cell to leave the cell and go do other functions in the body. So here's our Golgi apparatus. Again, you can kind of see the location. Here we have our ER. Here we have our Golgi apparatus. Again, here is our cis face that's closer to the ER. Here's our trans face that's a little bit farther from the ER and closer to the plasma membrane. Again, if you look at the Golgi itself, you'll see kind of these flattened stack areas. Again, that's what our Golgi is. It's kind of these stacks of flattened sacs. Our next one is a lysosome. Again, remember these are our membranous sacs or our membranous organelles, and lysosomes are small membranous sacs. Now, we talked about this earlier when we talked about transportation. So things like transcytosis, exocytosis, endocytosis. We talked about the lysosomes as they are a part of this process. And so what lysosomes do or are is they contain digestive enzymes that are formed by the Golgi. And these digestive enzymes basically come in and help to break down substances. So they're actually going to participate in the digestion of unneeded substances. And they digest the contents of endocytosed vesicles. So again, as substances are pulled into the body or into the cell, we have these vesicles that are formed with endocytosis. Those vesicles then form or join with a lysosome, and the lysosome releases its digestive enzymes into that vesicle. And those digestive enzymes basically break down whatever substance was in that particular vesicle. Now, peroxisomes are also membranous sacs, but they're a little bit smaller than lysosomes. These are pinched off vesicles from our rough ER, and a lot of these proteins are actually incorporated to serve as the enzymes of the peroxisome. And the function of a peroxisome really is in detoxification. So they are a little bit similar to lysosomes, except some of their contents are a little bit different. So again, whereas lysosomes are really for digestion and breaking down of the substance, peroxisomes use proteins and enzymes basically as a way to detoxify a cell or detoxify an area. So if we look at a little bit of a clinical view, we look at some lysosomal storage diseases. And these are typically a group of inheritable disorders, okay? We have this accumulation of incompletely digested molecules within the lysosomes. And so this actually allows for the mutation in genes of our lysosomal enzymes. For example, we have Tay-Sachs disease. In Tay-Sachs disease, we have a lack of enzyme that's needed to break down complex membrane lipids. And because we're not breaking down lipids quite the way we need to, or in quite the amount that we need to, we actually see that lipids will begin to accumulate within the nerve cell. And by the accumulation of lipids and fats within the nerve cell, that actually slows the function down until we slowly get to paralysis, blindness, deafness, and then oftentimes we get to a point where we often have death by the age of 4. So as we continue with the membrane-bound organelles, we look kind of at a collection of all of these organelles. And that leads us to what we call the endomembrane system. And the endomembrane system is an extensive array of membrane-bound structures. It's going to include the ER, the Golgi apparatus, vesicles, lysosomes, and peroxisomes. It includes the plasma membrane and the nuclear envelope, and is connected directly or through vesicles moving between them. And so what this endomembrane system does then is it provides a means of transporting substances within the cells, okay? So it allows us to move from, in this case, the nucleus to the ribosome, to the ER, to the Golgi, into vesicles, join with the lysosomes and peroxisomes, or it can actually join with the plasma membrane. So if we look at our mitochondria, now the mitochondria are also membranous structures. However, these are a little bit more oblong-shaped organelles, and they do have a double membrane. The mitochondria is really the site of cellular aerobic respiration, and so it basically provides us with a lot of energy, which is also why it's called the powerhouse of the cell. We have the complete digestion of our fuel molecules, and this allows us to synthesize ATP, and therefore the powerhouse pseudonym. We also have ribosomes. Now, we kind of touched on ribosomes as far as where they're located, or at least partly where they're located, but we now have to kind of understand what ribosomes are. And so ribosomes contain protein and RNA, this ribonucleic acid. We're going to see that ribosomes are actually arranged in large and small subunits. And when we kind of look deeper into the ribosome and open it up per se, we're actually going to see that the large subunit has A, P, and E sites. Our ribosomes are actually made within the nucleus, and they are assembled in the cytoplasm. Now we'll notice that a lot of these ribosomes are going to be bound ribosomes. And what that means is that they're attached to the external surface of the ER membrane. These particular ribosomes that are bound to the ER are going to synthesize the proteins for export. Oftentimes they're going to become a part of the plasma membrane, or they can serve as enzymes in lysosomes. Now our free ribosomes, these are actually going to be suspended within the cytosol. So these aren't directly attached to the ER, but they're going to be found randomly spread throughout the rest of the cell. And if the proteins or the substances that they're synthesizing don't fall into this category here, they're actually going to fall into this category then. And so all other proteins within the cell are actually synthesized in the free ribosomes. So here we take a look at kind of the structure of a ribosome. You'll see that we have this graphical representation. We have the large subunit here, and we have the small subunit here. When we join those together, we get the functional ribosome, okay? And so you'll notice that it has the E site, the P site, and the A site, and these are all going to be locations where we are actually binding our amino acids or bringing in our tRNA molecules as we relate to protein synthesis that we will get to here later this chapter. So now we have a handful of non-membranous bound organelles. Our non-membranous bound organelles, the first one is going to be a centrosome. And the centrosome is a pair of perpendicularly oriented cylindrical centrioles, and they're surrounded by amorphous proteins. The primary function of our centrosome is to organize our microtubules within the cytoskeleton, and it functions in cellular division. I believe you guys do touch on this in lab, but we will also talk a little bit about this again, I believe towards the end of this chapter. So here's our centrosome. Oftentimes the nucleus is kind of considered uh, the heart of the cell or the brain of the cell, if you will. The centrosome is oftentimes considered the center of the cell because, again, this is where we have our microtubules and structures that are involved with cell division. So we're essentially trying to divide the cell into two equal components. And so we kind of look at the centrosome as the center. Other non-membrane organelles really include components of the cytoskeleton. And the cytoskeleton is really exactly what it sounds like. If you break the term down into its component parts, we have cyto, and then we have skeleton. Well, we know what the human skeleton is. It involves our bones and provides us structure and allows for mobility. Well, in this case, it actually plays the same role, except now because we're not talking about the human skeleton, we're talking about the cytoskeleton, and cyto refers to cell. So this is basically the skeleton of the cell. And so what it does is it plays roles in intracellular support, organization of organelles, cell division, and movement of materials. Really, if you think about it, all of these functions then we can really translate to a larger scale onto the human body. Now our cytoskeleton extends throughout the cell interior and it anchors proteins within the membrane. And there really are three components of our cytoskeleton. We have microfilaments, we have intermediate filaments, and we have microtubules. And we'll discuss all of those. So our microfilaments are basically microstructures. They're the very smallest components of the cytoskeleton. These involve the protein actin. And so we have actin protein monomers that basically form into two twisted filaments. The functions are really to maintain the cell shape, the internal support, as well as can be involved in cell division. Our intermediate filaments basically are intermediate sized. They're a little bit more rigid than microfilaments, so they have a little bit more structure, a little bit more strength to them. Their functions are for structural support, but also to join cells together, so cell junctions. And then our microtubules are the largest components of the cytoskeleton. And they're going to be composed of the protein tubulin. Oftentimes you'll see that microtubules can be elongated or shortened as needed. And again, similar to some of the other structures, the functions of the microtubules are to maintain cell shape, for cell transport, but also microtubules are going to be involved in cell division. So here we get a chance to kind of look at all three different components of the cytoskeleton. Now again, we look at the microtubules, and those seem to radiate out from the centrosome, okay? You'll see the tan structures are the intermediate filaments. Again, these are a little bit larger than the microfilaments. They also had a little bit more strength to them. And if you look at the intermediate filament, you'll notice that they're basically just a bunch of strands that are twisted together. And what these resemble is, if you think about a cable, a metal cable that holds structures together, all metal cable is basically a bunch of metal wire that's twisted together. And by doing that, you actually significantly increase the strength of that particular groupings of wire. And that's what our intermediate filaments do. They basically take a bunch of these strands, wrap them together, and that adds a lot of strength to the equation. And then we have our microfilaments, which again are made up of these actin subunits, these actin monomers. And so we basically just grouped a bunch of them together and again took that essentially a double helix type model and twisted them together as well. So now we're not necessarily looking at the parts of the cell from an organelle internal structure, but what about some of the structures or the organelles that are found either on the surface of the cell or on the external structure? Well, that's going to lead us to cilia, flagella, and microvilli. So cilia and flagella are both extensions of the actual cell itself. They're kind of these hair-like projections. Now, cilia are literally hair-like projections that move substances along the cell surface. Okay, so they're not really involved in the movement of a cell so much as they're involved in the substances that are touching the external surface of the cell. Whereas the flagella, uh, when we talk more along the lines of a sperm cell, are longer and wider than a cilia, and they actually propel the entire cell. So they're involved more in cellular movement. Cilia are involved more in substance movement across the cell. Now, our last one is a microvilli. Microvilli are extensions of the plasma membrane. And what these microvilli do is they increase surface area. So by increasing surface area, you actually allow for a larger area of absorption into the cell. So if we look at microvilli here, again, this is all a part of the cell. These are all extensions coming off of the cell. And again, if you kind of look at this 3D shape or this 3D structure, basically you see where fluid can basically get down into all of these crevices through here. And by getting down into the crevices, again, it's allowing more surface area and it allows for better absorption. We oftentimes see these in the small intestine, where again, we're looking to absorb substances from the foods that we're eating. Now we're going to get into a few membrane junctions. Now we have this graphic here. These graphics basically represent all three of the different types of junctions that we are going to talk about. We have a tight junction, a desmosome, and a gap junction. And so we'll go ahead and we will touch on all of those. So our first type of junction is a tight junction. Now, tight junctions are going to have strands or rows of proteins that are going to link the cells together. They're going to prevent substances from passing between cells, and it requires materials to move through rather than between the cell. Desmosomes are a little bit different. These are going to be composed of proteins that bind neighboring cells, and they're going to act as rivets or spot welds. So what we're going to see is that with desmosomes, there's actually a little bit of give and a little bit of movement, whereas in tight junctions, again, there's basically no passing through. And then we have a gap junction. And a gap junction forms tiny fluid-filled tunnels. So we actually are using some of the channels that we had talked about earlier to create this junction. And it provides a direct passageway for substances to travel between the cells. Examples of each of these. So in a gap junction, you'll see it here, that we have cardiac muscle. So again, that's how cardiac muscle essentially gives us this all or none contraction. The heart either beats or it doesn't in a normal functioning heart. And that's because of the gap junctions that allow ions to flood into each cell, and it causes this flood of ions to move within the cells from cell to cell. An example of a desmosome is actually going to be our skin cells. Again, we have these welds where we have a little bit of a give, a little bit of play. It doesn't provide a complete barrier, but it does provide a relatively strong barrier. So that's going to be a desmosome. And then a tight junction, you have to really think about looking into the intestine and looking into areas where we don't want things to pass through, specifically within the stomach lining or even in the intestinal lining, where we have bacteria and protein, or bacteria and pH differences. If we have a break in that lining, You run the risk of bacteria getting into your bloodstream, causing sepsis. You also run the risk of acids getting out of the stomach lining. So again, that's where tight junctions within the intestinal tract really come into play. So I'm actually going to scroll backwards here. So if we look at this little section here, you get a chance to see each of these. So here we have our tight junction. And again, essentially, it basically locks everything into place and forms a tight joint. Our desmosome has these proteins that, again, almost like zipper teeth, where you can see that they can actually slide over one another, and that allows us to have a little bit of give and play in that particular junction. And then lastly, you get a chance to see the channels here, where again, we have these pores, this opening, where we're able to get ions to essentially pass from one cell, go through to the next cell, and so on and so forth. So that leads us then into our nuclear envelope and the nucleolus. Our nuclear envelope is kind of a double phospholipid membrane that encloses the nucleus. It separates the cytoplasm from the fluid within the nucleus, which is called the neoplasm. And then externally, it's continuous with the rough ER. Now, as we exit our nuclear envelope, or we exit the nucleus through that nuclear envelope, we're actually forming a nuclear pore. And the nuclear pore are these open passageways formed by proteins within that nuclear envelope. And these nuclear pores allow the passage of large molecules into and out of the nucleus. And if we think about some of the large molecule structures, one of the largest structural molecule in our body is a DNA molecule. And DNA is actually stored within the nucleus, but as we go through the process of trying to create a protein, we actually create what we call a messenger RNA molecule, or an mRNA molecule. And that mRNA molecule actually exits through the nuclear pore, okay? And that leads us right to our ribosome. We also find that ions and water-soluble molecules are able to pass through our nuclear pores. Now our nucleolus is a dark staining spherical body. It's not membrane bound, so we don't find it in the membrane or on the membrane or enclosed by a membrane. It is composed of protein and RNA, and it does help to produce the small and the large ribosomal subunits. The nucleolus is not found in all cells. However, we can find more than one of them in nerve cells due to the production of many proteins. But as we said, it's not present in all cells, and the example would be a sperm cell, because basically in sperm cells, no proteins are produced. And so here we get a chance to see our nucleolus. So here is our nucleus. This outer blue layer is our nuclear envelope. Here you can see these dots here. Those are going to be our nuclear pores that basically are these invaginations in and around the nuclear envelope. And then all the way within the nucleus, you see our nucleolus, okay? And again, so that's kind of our structure of the nucleus itself. So now we're going to transition from essentially cell components to now we're going to start looking at our DNA. And we'll start getting into some of our physiological processes as we move along. And so as we start to look at our DNA, our chromatin, and our chromosomes, we look at the DNA specifically. Now, DNA is actually housed within the nucleus. And if we look back or think about the DNA structure, it's composed of these repeated monomers that we already know as nucleotides. DNA also, specifically when we talk about nucleotides, DNA has what we call deoxyribonucleotides. Now, each deoxyribonucleotide is composed of a 5-carbon sugar deoxyribose, a phosphate, and one of our 4 nitrogenous bases. And these nitrogenous bases, again, are going to be adenine, cytosine, guanine, or thymine. So A, C, G, and T. Also, if we look at our DNA, we talked about how DNA and RNA are formed and how the nucleotides are formed as far as bonds go. And they're linked by our phosphodiester bonds. And you can refer back to a few lectures ago when we talked about phosphodiester bonds. We actually had that image or that graphical representation as we were discussing these components. I believe in chapter 2 or 3. Each molecule has 2 complementary strands of nucleotides. So again, this is a double helix molecule. And these double strands basically form like a spiral ladder. And so the sugar and the phosphates really are going to form the struts of the ladder, whereas the pairs of the nucleotide bases are going to form the rungs of the ladder. Now, they're not directly connected to each other as far as the 2 strands go, but what they are connected by are hydrogen bonds. So the complementary strands on either side are really connected by the hydrogen bonds that are formed between the nucleotide bases of either side. Our DNA molecule houses most of our genetic material of a cell and really gives us all of the instructions or the blueprints on how to create or what we need to do to form most of the proteins of our body. And a human actually has 46 separate double-stranded DNA molecules. And so we'll take a look at how these are actually formed in one of these next graphics. DNA is also a very good way that we can identify our attendance today. So attendance code word is actually going to be DNA. I think that'll just fit in just right for today. So DNA is also a double helix that's wound around the nuclear proteins that we call histones. Now, as we take this winding and we take these histones together, these are actually formed or called nucleosomes. Now, when DNA is not dividing, it's in a form of finely filamented masses called chromatin. But when it is dividing, it actually forms a very tightly coiled mass. And this mass is actually called a chromosome. So if we take a look here, you'll see that this chromosome kind of looks like an X almost. And so these are basically the components of a chromosome. You have this DNA molecule that's basically all tightly formed, bound together. And that's how we're able to start to divide the cell and give each cell equal or the same amounts of DNA. Otherwise, it's kind of this uncoiled, if you will, structure. And here we get the chromatin, okay? But now if we get down to the detailed components of it, we get the histones, basically stacks of 8 here, and then we get the DNA molecule really to wrap around those histones essentially twice. And then it goes on to the next group of histones and the next group. And this gives us essentially our DNA strand into this wound chromatin, and then we tightly compact it into a chromosome. But now if we look at the strands by themselves, again, you'll see that here is strand 1, here is strand 2, but then we have these bonds between them. And this is our nucleotides, basically again looking at G's bind to C's and vice versa, and A's bind to T's and vice versa. And so this is kind of our structure of DNA and chromatin. So as we look at the DNA and our genes are really these segmental units of nucleotides. So we have this long DNA strand, But within that DNA strand, we have small little sections. And each of these sections are basically considered a gene. And each individual gene provides the instructions for the synthesis of specific proteins, or sometimes a specific protein. And within that gene, we're actually going to find what we call a start signal or a promoter region. And we're also going to find a terminator region, or what we would consider a stop signal. And this basically indicates the beginning of the gene and the end of the gene, or another way to say it, the beginning of the coding for a proton and the end or the stop point of the coding for a protein. And we'll actually stop here for today because the next part of our lecture actually gets into one of our physiological processes. And so we'll start our next lecture with that one.